Hello, everyone, and welcome to today's seminar in psychology. And we are honored to have two distinguished researchers from Europe, Professor Prof Forstmann from the University of Amsterdam and Professor Miletić from Leiden University. They will share their research with us today. The first talk is from Professor Forstmann. She earned her PhD from the Max Planck Institute for Human Cognitive and Brain Science, and is now a professor of neuroimaging in cognitive neuroscience at the University of Amsterdam. Her research focuses on how the deep brain or subcortex support mental functions by combining mathematical modeling with neuroimaging technique, including ultra-high field 7 Tesla MRI and postmortem study. She has greatly advanced our understanding of how the subcortex influences cognition and behavior. Her work has been published in several top-tier journals and earned grants and awards from the European Research Council and the Netherlands Organization for Scientific Research. Today, she will talk about joint modeling of the deep brain's form and functions. Please join me in welcoming Professor Forstmann.
Sorry, Juan. Thank you very much for this kind introduction, and thank you very much for having us here all the way from Amsterdam. In fact, before I start, I'd like to share some impressions of our past days here in Taiwan. In fact, at the Mount Ali. So we spent 2 days, a lovely sunrise with our dear friends, Shulan, of course, you all know, Professor Xie, and Professor Wong from Shai. And yeah, you see, we enjoyed it greatly and had fantastic impressions. So thank you very much for giving us the opportunity to experience Taiwan and yeah, to just have the opportunity to meet such lovely colleagues and friends really by now. So it's my second time to Taiwan. I have been here already to your university 10 years ago, and it's just a pleasure to come back. Yeah, so in my talk today, actually, before I start, I'd like also to introduce myself very briefly. Already you heard a little bit. I did my PhD in Germany. I am originally from Germany, and I did this with these lovely people. You may, in fact, soon see Irin Koch, who will be visiting your university as well. He was my PhD supervisor next to Marcel Brass, and also Yves von Cramon. I don't know if I can I close this? Does it say something important? And then about 20 years ago, I moved to the University of Amsterdam. That wasn't really a planned step, but it happened. And so I only wanted to spend a little while abroad, but well, now 20 years later, I'm still there, which is lovely. And so Richard Ridderinghoff was one of my supervisors. Some of you may have read papers of him. And I then also met Erik Jan Wagemakers, who is a mathematical psychologist, and with whom I started to collaborate for a long time, and with whom I started to develop the so-called model in the middle approach, or model-based cognitive neuroscience. I should say that this is truly not a new approach when we introduced this. There have been people like Jeff Schall working with non-human primates and O'Doherty, who basically also have used this type of approach earlier. But to introduce you to this, and this really builds the backbone of all the research you're going to see today, is that we wanted to go beyond classical cognitive neuroscience research. That means that you basically collect some fMRI data together with maybe some behavioral data, and then you interpret pretty much like these brain data, functional brain data images. But in this model in the middle approach, what we're doing is we use so-called formal quantitative models, and I'm particularly referring, for example, to evidence accumulation models or the depth diffusion model, for example, that helps me to understand modeling behavioral data, latent cognitive processes. So processes that I can't directly observe by means of reaction times. And I'm going to explain in much more detail what I exactly mean with that. And in fact, in the second talk by my friend and colleague, Prof Miletić, you will see he will give much more insight how exactly these models work and what you can do with them and how powerful they are. But in this approach and talk, I'm going to focus on the intersection between using these formal models and estimating parameters per subject, which I then use, for example, in my imaging data as a correlation or as a covariate. So I want to bring this latent mechanistic understanding together with my brain imaging data. This approach has led to many beautiful collaborations in the past. And here you see many members of our lab, former members who have used that approach. And I'm really grateful to have had the opportunity to work with these people who have, some of them moved on. So you immediately recognize this person here. He started as a research assistant and now he's assistant professor in Leiden. But also others. Maybe some of you remember Wouter. He has spent some time here. Yeah, he looks like a baby. I know he's grown. He was here. He was a PhD student of mine, and I think he did some classes here to teach diffusion imaging, and collaborated with Shulan. But also others. For example, Matthias. He's now full professor in Norway at the University of Tromsø. So many people who really enjoyed science, used this approach and just basically enjoyed academia and also then made their own career in science, which I'm very happy about, that this framework kind of provided a little bit of a home also for then moving on and using it in many different, different research topics. So, but to give you a brief outline of this talk, first I'd like to talk a bit about the deep brain and what exactly this means. It's a term that not many maybe have heard of, and it's not really a scientific term if you want, but it describes regions that are lower than cortex, so beneath cortex. I'm going to show you exactly where and what I mean with this. Then I'm going to present to you a bit joint modeling of brain and behavior. And this is a fairly new approach where we try to come up with models that jointly basically bring together parameters of latent cognitive processes and brain imaging data. But I'm going to show you exactly how this works also by means of a concrete example. And then finally, if we have time, we'll see, just get into some future research directions that I think could be valuable given what I'm showing you here today. But maybe you also have different ideas. So I'm very open for discussion. Also, if you have questions, I'm going too fast, anything's unclear, just please raise your hand. I'm very happy to do this much more interactively and not just talking to you now for 45 minutes. So please don't fall asleep. So if you have questions, please just go for it. Yeah, so this is a slide that is a representation, it's really a schematic mid-sagittal of the brain. So, right, we're looking here at the brain. Basically, if I cut it in half, you're basically looking in the middle in this section. And it's an image that you will find in many journal articles still to this date. And what it basically shows for now is in colors, different fields and cortex. And this is just a simplification because researchers have found out that there are approximately 180 distinct cortical fields. So that means that there are 180 areas that are basically distinct and eventually also code for distinct cognitive functions. But what is striking, and this is something that we were sitting around in Amsterdam with the lab, we were a bit bored, we looked at images and all of a sudden somehow this idea came up, hold on, there is not really a black hole here, right, in this brain. That's unreasonable. But I can assure you, if you look at connectome studies, if you open articles right now in neuroimage or imaging neuroscience, you will see a lot of representations that just simply say, this is black. And I can assure you, it definitely is not. It's anything else than black. There's not a void. This is not just like a large ventricle. There are ventricles, but more so, what we counted is that there are about 455 unique nuclei residing deep down under the cortex. So 2.5 times more than cortical fields. Sorry, and this is quite striking because as a cognitive neuroscientist, what I was really curious as a next very concrete question is, if I do my imaging analysis, I have collected fMRI data, assume, right? And I would like to investigate now these 455 structures. How many atlas maps can I actually download and register my data in order to inquire what's happening there? And so the striking number of only 7% came up. So if you use, for example, standard software like FSL, SPM, that are really standard software analysis packages, if you do fMRI analysis, this is basically what you're left with if you use all the atlas maps that are readily available there. So what I'm getting at is pretty much this, and that is that I'm arguing that this deep brain, the human subcortex, so everything underneath cortex is pretty much terra incognita. So that means uncharted territory. We know it exists because anatomy tells us people have dissected brains, so we know these regions are there. But what I'm arguing is that they are basically uncharted. So no one has taken the effort in order to bring them, for example, in a comprehensible atlas, so that you can use this to inquire about structural properties, functional properties, and so forth. So basically, what I'm showing you here is a world map, obviously, and only about not even entire Europe, but we could maybe also point to a little bit areas in Asia, is mapped in red, but the remainder, all these gray shaded areas, are unmapped territory. Again, areas are known. I'm not arguing about finding out new areas, but redefining the brain and completely, not at all. But what I'm saying is there's a huge knowledge gap. You can spend your entire research career, if you want it, just on, let's say, a tiny little bit of Italy, right? Or a tiny little piece of Taiwan. And you'd be busy for the rest of your life. I spent about 8 years on I don't know how big it is, but I would say, let's say, as big as my hometown in Germany. Yeah, 8 years, and I'm still not coming to conclusive results. So just looking at that, I can tell you, you will have a bright future. You can be very, very busy by just even take a dart and just throw it against this map and then start to investigate that, not of course a geographic region, but a brain region. You will be busy. Yeah, I'm still amazed that this is so under investigated, and I should mention importantly in the human situation, because what I learned from researchers are using animal models, and here I'm talking about, for example, rodent models or non-human primates, we see that they are facing the reverse problem. And that is quite understandably. So for example, in rodents, the subcortex is much more prominent. And so researchers have spent much more time investigating the functional role of subcortical regions, given that cortical regions in the rodent situation are very, very small. So in ratios, if you want, cortical, if at all they have cortex, that also stands out to be argued. It is a fraction of the mass of brain that the rodent is made of. So these people say, well, subcortex, well, boring. We know everything about it, and I believe that. I understand. So we need to understand cortex. However, as I said, in the human situation, completely different. And I'd like to explain a bit why I think this is, because it's not intentional. It's not that, let's say, human researchers doing human research, like functional research, decided all of a sudden we are all going on cortex. That's certainly not true. I guess cortex is so fascinating because it gives rise to higher cognitive functions. There's no argument about it. And eventually it might differentiate us from the animal situation. And I think that kind of intrigued many researchers to say, let's just focus on cortex. But I think another one is a more methodological one. And here I'm going to show you a quantitative meta-analysis. These are results of the study. So what we did is a former PhD and postdoc in my lab, Max Köken, he harvested the literature. And with that, I mean not just 10 or 20 or 30 papers. What he did is he did a massive search, an automatic search on PubMed and other databases that hold literature. And he harvested over tens of thousands of articles that held information about terms of subcortical nuclei, for example, thalamus, striatum, or putamen, in association with cognitive terms, for example, decision-making, consciousness, perception, attention. And the idea was to test to what extent and which regions are most often mentioned with respect to a cognitive term. So right now, this doesn't represent which cognitive topic is really related to what region. What I'm showing you here is more a bias towards larger structures in the subcortex. And I think that immediately gives some explanation why there is such a large knowledge gap. So what happens here is, and that may be very much driven by current methodologies that we have for non-invasive investigations, that people simply just go for certain regions, like the striatum. Whom of you does know the striatum, have heard of it? All right, it is, I can promise you it's a very important and prominent deep brain structure. Who has heard of the thalamus? Yeah, more people already. So again, a good sign. So, you know, even if you haven't studied the brain, you know about the structure. And that's exactly what's happening here, what I'm arguing. People tend to basically report things or structures that are larger, but that eventually maybe also be more known in the literature. But that doesn't reflect or give clear insight really what's happening down there and a clear understanding of brain structure and function relationships, let alone that many of these 455 regions aren't mentioned at all in the literature. And that can't simply be true. There is clearly a problem. But I think another clear issue why it is so difficult to image and understand the deep brain in the human situation is that many of these nuclei are extremely small. So you'd be happy actually ending up with a nucleus that's as big as maybe your pinky nail. And already my pinky nail is small, I would say. And so many structures are even much smaller than that, and also in volume. And their shape is also sometimes really quite weird, lens-shaped, all sorts of things. Another issue is that they, as you can see in this slice, they lie in very close proximity. Really different than in cortex. Sometimes you can't even differentiate between different nuclei bordering each other. And we also see that some of them have very high iron content. So I didn't know that when I started this research, that every one of you guys, including me, we carry heavy metals in our brains. For example, iron. We also carry, for example, copper. We carry a lot of things which I didn't know and which are really important to know if you do functional or structural imaging. And I'm going to show you why this is extremely important. It is also important when we think of diseases, for example, Parkinson's disease. We understand now that iron accumulation eventually may lead or is leading to the progression of motor diseases such as Parkinson's disease and gives rise for motor tremors, for example. So there's a very interesting link also to the mechanisms and accumulations of these heavy metals. So to overcome all of these biases, to actually give it a start, to give it a try, to better understand and close this knowledge gap of the deep brain, we started to collect a rather large database. You may think, 100 subjects, and I agree with you. If we look around in the world, you also find databases that contain 1,000 subjects or even 10,000 subjects. Absolutely. But mostly these databases do not have high resolution scans. So in this database, what we try to do is collect data with a rather large age range from 18 to 80 years using 7 Tesla MRI. Whom of you have heard of 7 Tesla MRI? Right. So who of you has heard of magnetic resonance imaging, MRI? Right. Okay. So these machines, stop me if it's really gonna become very unclear, but these machines are big machines, very big machines. And basically a subject is just put on a tray and they're basically put into a large magnet. So it's a hole, and basically you are shoved into this with the reason that this machine delivers images of the brain. And dependent on the magnetic field, just very briefly in terms of physics, you can take images with more or less resolution. That means these images can become more and more detailed the higher the magnetic field gets. So if you, for example, break a knee or you have something going on in your head, most likely you go to hospital and you're going to be shoved in one of these machines, but mostly they are lower field. And that is perfectly fine because still they deliver insight. But what I'm talking here about is a machine that is still very research oriented. There are clinical uses now as well. And I think you do have a machine like that in Taiwan as well, a high field machine. And the beauty of these machines is they look exactly like other machines, but because of this high magnetic field, we are getting much more detailed images of, for example, brain structural features as you would get on, let's say, more common 3D MRI machines. But what I'd like to show you here is that, again, we collected all of this data of these 100 subjects. In fact, we had 2 waves, a first wave and then after a year, second wave. And we used a sequence. It's called an MRI sequence. It's just the settings with which you measure the brain, which rolls out different contrasts. And that's exactly what you see here. Basically, it rolls out in one go simply different types of contrast or sequences that roll out different features of the brain. And what I completely mean with that, and that is very intuitive, and I think everyone will immediately understand, if you look at this image and you compare it with this, right? And you, for example, focus at these dots that you see here. This is the red nucleus, it's called, doesn't matter. If you look at this image, right? Does anyone see the red nucleus there? Great. Thanks. No, exactly. You, for example, see certainly globus pallidus interna externa, you do see some contrast here. So some features appear also in this contrast, but others are completely lost. And so what I'm arguing is that you not only need really a very high resolution, so strong magnetic fields in order to resolve the deep brain, but you also need to take care that you acquire different contrasts as to get to different features in the brain. Because else it's completely lost. You have no insight and you can't see it. So this database served us in the first instance to create an automatic segmentation algorithm. And I should say everything that I'm presenting here today is really available. So should you be excited and should you in fact decide today I want to become a neuroscientist, you can just go ahead. Just download all of this tonight and start to play because everything is there. You could even start to conduct your own study if you like, because everything is readily available. You don't even need to leave your email address. Because the reason is that we are really eager in closing this gap. We're very eager and we know that we need help and that we need collaborators, that we need scientists who become more and more interested in mapping the human subcortex, because this automatic algorithm was developed in our lab and it is quite powerful. It can automatically segment already 63 individual subcortical nuclei. But as I told you, we're looking at 455. And we once just sat down because we got a really funny rejection from a journal saying, what is this? Why do you only do 16 structures, right? When you argue 455, so do the job, right? And so we sat down and calculated. And I replied to the editor and said, look, dude, it would be 455 years, right, from now, if we were to do all of them the way we actually started doing it, because we did it very, very carefully, validating our results with raters. So a very lengthy procedure. So getting an atlas of one nucleus takes about 3 months. So you can calculate, and that's just maybe one structure. And there are many structures that are much more complicated. So this is not just a simple task for research assistants or so to do. But as I said, it's a start. Other labs have joined efforts. There are groups that also are very clearly eager to map other regions, also cortical regions, which are implemented, for example, in free surfer. So I'm not saying we're the only ones, definitely not. So it's starting, and I'm very happy about it. But it's a long way from here until we basically really have maps of all of them. Yeah, of course. Yes. Yeah. Yeah. Excellent question. Yeah, excellent question. So we benchmarked, it's actually Pilou Bazin. He was a former senior scientist in the lab. He's still associated with us. He did this work. He programmed this algorithm up, and he also benchmarked this algorithm on 3T HCP data. So that we were very careful, and we wanted it to work also on 3T data, knowing that, of course, most researchers will have T1 weighted images. The only objection to this is, in fact, if you only have one modality, that is mostly people scan only T1 weighted, right, as a structural reference space. And that is a problem for this algorithm. So preferably what you would have is at least another contrast, for example, T2 star weighted, but with that comes, for example, QSM. And then this works much better. But it's also no problem. For example, if you don't want to work from your individual data that you scanned yourself, you can also, for example, co-register atlas maps, probabilistic atlas maps that were derived from these 70 data. And then you can investigate always, of course, yourself, visually also, does this make sense given your resolution? Absolutely agreed, right? Some of these structures become so small that eventually a 3x3x3 doesn't make any sense, to then pretend you do LC research or something, right? But that is left, of course, to the consideration of the researcher themselves, and being very careful of what and what cannot interpret it given your own data. But what I'm arguing, what I'm saying is that the algorithm does work on it, right? If you have at least two contrasts. Yeah, that was really our idea, not to make it so specific that you need exactly that sequence and that parameters or whatnot. It is benchmarked, it is validated on other data sets. Yeah. But yeah, to go even one step further, and this is really a little bit of a detour that I'm taking you on now. But I hope I bring this all back together in one framework. And in fact, the idea is here to understand cognition, right? I'm not an anatomist. But as research goes, you start something and you realize one boundary after next boundary. And so you have to decide, do I going to go push through or do I stop here and know that there's a problem, but pretend that actually there isn't. And I just, do go the other direction and just don't look at it anymore. So I think I pushed through, definitely also because I met Anneke Alkemade, and that was essential for me. She is the anatomist in our lab. She's a senior, she's an assistant professor. She is trained in anatomy, and I would have never dared to build what I'm showing you now without her. So this is really work in collaboration with her and driven by her to make this happen. So we realized that even in vivo, so if we scan living human subjects, even in these high fields with sub-millimeter resolution, we can go down to 0.5 millimeter by now, even higher, we will still lose information. We will never get to the level of the tiniest nucleus. And even with this approach, I'm questioning whether this is possible. But so what we decided to do, and it was actually, so often also projects are born in my lab out of curiosity, out of fun, out of challenge, out of let's make this happen. While we know this can really go very, very wrong, but it's just fun. And so Anneke and I thought it is actually greatly fun and interesting to take entire whole dead brains. So that means we went to the anatomy department in Holland, in Maastricht, and we collected entire heads. So basically heads that are basically cut off from the body at this level approximately. So these are body donor subjects. They donate their entire bodies for research. And we started to collaborate with this anatomy department in order to collect these heads. And so we brought these heads to Germany, and we submitted them to a 48-hour scan protocol. And that is possible, obviously, because dead people don't move, right? I couldn't do this with a living subject, obviously, right? 10 minutes and you're done, because we're talking here about 0.5, 0.3 millimeter resolution, right? This is, I can't even show how small this is. And if you move, then this is all the images lost. So this provided us with a unique opportunity to collect incredibly beautiful data. But then, in fact, Anneke wanted to go one step further, and we brought these sets back to Amsterdam, and then we started to cut these brains on a macrotome. So we cut them in 200 micron thick slices. This is, again, incredibly small, and the entire brain was cut into approximately 800 slices. We did this because we said you have tissue, and you can stain this tissue. So you can work really by putting color on the tissue, and with that you start to see cells, you start to see neurons. So you start to see a detail that I can never resolve with MRI. But of course, we want to learn now. I want to bring this all back into my research area, which means somewhat cognition. And this is a bit removed. Bear with me. I'm gonna come back to the living world. But in order to accomplish this and do this, we need to bring this all back into 3D space. I cannot work in 2D with my living brains. That makes no sense. So we made pictures of all these slices, and then we constructed the entire cut brain into 3D. And that is exactly what you see here. So this is one of these specimens. We have by now collected 6 brains that underwent this procedure. And you see here this 3D reconstruction. You see even the stripe there. This is in fact an incision when the brain was extracted out of the skull. Here you see the different stains for neurons, for cell bodies, for different types of features in the brain. And this is all co-registered then with MRI. And that is very important because now I can align and I can co-register this high-level detail with my in vivo data, right? So you can, again, take this data, you can bring it in the space of your 3D data or whatever you have, and you can see on that level the cell body distribution in that region based on the specimen. Again, you can download all of this, what I'm showing you, but be aware if you download this data set, you better have a large server or really a large external HDE because this is very, very big. So don't try to put this on your laptop. I think this would give you a headache. But it is nicely formatted and really no problem at all to put somewhere and then play around with it. You don't need really extensive knowledge at all in order to do that. So that is really for the atlasing work if you want, and that's kind of ongoing efforts, obviously, ongoing atlasing efforts that is being done and done really in Anneke's team by now, in the anatomy team. But I'd like to turn now back to the living situation and also some methodological problems that we ran in there. So sorry that half of my talk is just talking about problems and trying to solve them until I come to something that where we really make use of all of that, what I'm showing you here. But this is another issue that we ran into and that I, as a supervisor, I really should have known, and I did not when I started this. It's a project that one of my PhD students, Gilles de Hollander, about 8 years, ran into. He collected the first 7 Tesla fMRI data that we ever collected at the Max Planck Institute in Leipzig. And we were very naive, I should say. I was very naive. I was thinking, okay, great machine, very expensive, fantastic data must come out, must work. And must work by just basically, increasing voxel resolution. That must work. Great. So we did that. And Gilles came back and said, did not work. And I was like, yes, must work because very expensive, right? And great machine. Yeah, exactly. Super stupid logic of a supervisor, but that's pretty much what I did with him. So we played that game for really certainly 3/4 of a year. Gilles, did not work. Birte, must work, did not work. Okay, great. So at some point I started to listen to him and happily I did. Very smart student and really persistent because basically what he did is what he wanted to tell me constantly is, Birte, I'm fishing in noise. We did all of it, but it's still noise. There is no signal. basically what he was telling me is that he was looking at these types of images. So here you see a heat map, and the more red this is, like dark red, the less signal. Pretty much really no signal. And I'm really still so appreciative, because he could have just, by means of statistics, somehow produced results, no problem, right? Everyone can do that. You can create false positives, you can write a paper and hop a case, say 7 Tesla, woohoo, it was great, right? But it was really not great. And the reason is again, and this comes back to the heavy elements that I told you about in the beginning, iron. So what happens is in certain regions of the brain, certainly deep in the brain, not in cortex, but deep in the brain, and in particular in a region we were interested in, this region has a lot of iron, and I just didn't know that. And I also didn't know that if regions have a lot of iron, you need to work harder in order to get signal, because basically what you're seeing here are decay curves. You see basically signal, and the speed with which the signal in a particular region just vanishes. This is normal, this happens. And so we need to adjust certain times to catch it. And so basically what we did, we set the time to this dashed line, which I thought prima, because everyone is doing it. And in fact, everyone is doing it. If you read a journal, if you open an article right now, you will exactly read this number, meaning 22 to 25 milliseconds. And this is the so-called echo time. Doesn't matter for you guys, just important. Never, ever, ever use settings from a vanilla standard fMRI experiment if you want to go deep in the brain. And if you in particular want to measure iron-rich nuclei, because it's going to be, you just measure noise. You could also just have not done that. So you see, this is our interesting region. This is the decay curve of this region. And you see that all signal is already gone by that time we measured it. And that's exactly why he came back and said, Birte, no, yes, maybe. And I'm not even saying really yes, but in comparison to the other settings, definitely better, and definitely something that we can now really work with and do functional experiments with. I should also note this is really only a problem if you go to high field. If you do lower fields, I wouldn't be able to say how to measure it as of yet, because we never really compared it. We really did a lot of quantitative comparisons to get to this, the settings. But my hunch is that also there you would be very, very careful and advised in order to run some pilots to explicitly test how much you get out of it. And also just maybe more for the experienced ones, you would think, okay, well, maybe it's much more efficient not to only set this value, one value, but maybe have a range of values, right? Have a multi-echo sequence. That is not beneficial. Steven did a lot of work in that direction. It doesn't help. What is really helpful is if you really shorten these times. And I can tell you, if you still think cortex is awesome, you're always safe, because cortex is super forgiving. So whatever you fiddle around here in terms of these values, you will always get a signal in cortex, no problem. Yeah, so equipped with all of this now, in fact, we felt, okay, we're really now ready. And this is now about 5 years ago, to study the deep brain in a meaningful way, in a mechanistic way. And this is what we set out. So we speculated that eventually there are different networks. Obviously, one is relating motivational information, one cognitive and more motor-related information. What is crucial here for our questions were these different subcortical nuclei that are involved, giving rise to these types of functions. So this is kind of the gross framework in which I designed or we designed several experimental paradigms to tap into these different psychological processes. And so this brings me to the second part of this talk, and this is going to get a little bit abstract now. And so Steven will explain in much more detail how exactly we do behavioral modeling. But just to give you a gist, just to give you an idea that we're not just computing classical vanilla GLMs for our imaging data, but still we want to bring together behavior modeling data and imaging data in a joint framework. And so you've seen this. And in fact, what we're trying now here is, instead of unidirectionally informing our brain imaging data, what we're doing now is we're building one joint model of behavioral data and of brain data. And this goes beyond simple correlations. We're really kind of going away from this now. It's also not just simply a regressor that you add in your GLM. And I'm going to show you how exactly we're doing this right now. So first of all, very briefly, and again, you get a much nicer and more informative introduction to these types of models in a second. But this is a so-called evidence accumulation model. This is a model that is very powerful for behavioral data because it allows us to segment basically different latent processes while making a decision. So obviously, for example, you do an experiment, you sit in front of a computer and you press maybe left and right responses, right? So you get reaction times. You also get error rates. You get entire distributions. And these distributions contain information of a whole lot of different latent processes. For example, very intuitively, you need to encode the stimulus, right? There needs to be time to just simply look at what you need to work on. And then there's, of course, also a motor time, so an execution. You need to press a button. And these are processes in the periphery that take up time, up to 200 milliseconds, maybe more, that are all confounding your reaction time. And I'm not interested in this stuff. This has nothing to do with the decision process I'm really actually after. So preferably, I just want to get rid of it. And so what I'm doing in this model is I'm estimating these times, and they can be captured in one particular parameter in this model, so-called TER or T0. Basically, I'm treating this as a dustbin. Like I'm dumping it there and I'm getting rid of it. Because what I'm really after is the decision process. So the stuff that really happens in the brain, right, on a latent level, that's what I'm after. I want to know when exactly and how exactly this happened, how you made a decision. And so in this model, it's actually very, very parsimonious, a very simple model. You basically have as many racers. So here, a left response racer, a right response racer for as many responses. If you have 10 responses, you have 10 racers. And so the simple idea is that the decision process starts randomly choosing from a distribution. And as soon as this arrow hits this threshold, so-called response threshold, a response will be executed. That's all there is to it. So the decision is really defined by a parameter called drift rate and a parameter called response threshold. For now, can be made much more complicated. You will see different scenarios in Steven's talk. For our purposes, for now, for introducing this is all you need to know. And I'm particularly interested in this response threshold parameter. Without further detail, just believe me, for now, for the experiments, I am. And I can selectively manipulate it. And with that, I mean, I can also get for my subjects, I can get a value of this response threshold. And that's what I'm after. So we use this model in pretty much all of the upcoming experiment that I'm showing you. So that's really, let's say, the model that we use for behavior modeling. On the other hand, I'm going to go into this. On the other hand, for now, sorry, for the modeling of our imaging data, we still use a classical GLM, a classical general linear model. And that means that we're basically estimating a beta weight coming from the time series that we collected while people lay in the scanner and basically do a task. So we can log the brain activity, if you want, towards cues or different events that happen during a particular trial. So this is really standard model to analyze fMRI data. And so we need these two models, so the race model on this side and the GLM on this side, now to get individual estimates for each individual for behavior and for the neural data to then link via a hyperparameter. This is, for example, a multivariate normal distribution for now, to basically link this in a hierarchical manner with each other. So this goes beyond, again, a simple correlation, because we're basically using a hierarchical Bayesian approach here as to bring these two estimates of behavioral and of neural realm together. It may sound all very abstract to you right now. I totally understand. I'm going to show a bit more concrete example in a little while, and also maybe break it down by means of this matrix, which I hope makes this all a bit clearer, because how exactly the joint modeling works is something, there's no time to discuss this in detail here right now. It would need an entire session, but I'm sure Steven is much more the expert here than I. No, we don't have time for questions afterwards. Well, you can send email. We need to go to the temple afterwards, so we are on a strict time schedule. But you are very welcome to always send emails and questions if you like, if something is really, if you're curious, if something is unclear. But for now, I'd like to focus you on this matrix, because this is the representation of results that roll out of this joint modeling framework. This looks different than what you may be used of fMRI research by means of like a heat map with activations on top and stuff. What we're doing here is we're creating different matrices. And one is, in fact, a behavior-behavior correlation matrix. One is a brain-brain correlation matrix. So this is really the correlation between regions of interest. We have 16. In fact, this can scale up to as many atlas maps now we created in the first part as we wish. You can imagine this matrix can become incredibly high, right? Like large. For now, it is rather small because we started only with a certain subset of regions. And what is most important, and I'd like to focus your attention on, is this red rectangle, because in this rectangle, it's equivalent to this, clearly, is now the brain behavior relationship by means of this joint modeling approach. And that is really all I'd like you to focus on when I'm showing you some results now. Because these ones are interesting for many different other reasons, and these are as well, but for now, just disregard them, just that you kind of get an understanding how results now from this framework will look like. And before showing you actually the experiment we ran and also the results, I think it's always extremely important also to mention why a certain new framework like joint modeling could be advantageous. Because often still I read papers or I review papers from my peer colleagues, and I'm often really very impressed about the creativity of analysis that people come up with and the sophistication. But at some point I realize, why did you do that, right? Why was this necessary? I mean, it's cool, but science should be fun and cool, sure. But I mean, there should be a rationale why you're doing it, right? Not just for the coolness or to show off that you know statistics. So what I'm getting at is you need to actually clearly have a rationale and indicate why is this the way forward in order to answer the question at hand, and why couldn't any other methodology actually do that, and in particular an easier one, right, that someone can access. And so Nick Stevenson, he's a very, very excellent PhD student in his last year right now in our lab. He is, I would say, rather mathematician than actually cognitive neuroscientist, and he lost, or he's very ingrained in mathematical psychology, and he developed really the backbone of this joint modeling framework. And he did a lot of simulation studies, which are also published, and also recovery studies, very careful establishing why joint modeling has advantages over simple correlations or other approaches. And I think one very strong argument is that it provides an explicit reciprocal link between different modalities. And here I'm not talking only about behavior and brain imaging, but I mean, you can also think about behavior across tasks, and you can think about behavior and structural imaging. In fact, you can replace all these different inputs by whatever you can think of, because it relies on this hierarchical structural distribution that then is linked at this first stage by this hyperparameter. Also, he could show, very important, that it improves statistical power over simple correlations. There's a lot to be said also why using a multivariate, so that we can go into much more detail, but for now, just argue for improved statistical power. A very important, I thought also that it's generalizable, and we have recoverable principles. And finally, it provides an estimate of the uncertainty of the correlations. And this is, of course, also due to the Bayesian framework, something that, we absolutely endorsed. But so, very abstract, maybe very theoretical. In the last few minutes, I'd like to give you some insight into a concrete example, how you could use this approach. And this is work now by Steven, and Steven again will explain this in much more detail, more on the behavioral level, how the joint venture between reinforcement learning and speed accuracy trade-off can be modeled and in fact manipulated, and also which brain areas in the end actually are elicited due to these manipulations. What we see, and that is really striking, we are looking at literature in the reinforcement learning realm and also in the speed accuracy realm of more than 30, 40, 50 years, and they're completely separate. Yesterday, I used the example in Chai. It's pretty much as if someone developed something in the Netherlands and someone else developed something in Taiwan, and these people never talked. There was also maybe no internet. You could argue many reasons why this never happened, but it is a bit awkward, I should say, in particular since we have internet and we also have this for a very long time now, and still they didn't somehow get together. But now we do. Now we're here, and I think it is really beautiful. In particular, also Steven's work. There are also other groups that I think a couple of years thought this is awkward. Why isn't this thought together? Why is this treated so separately? Maybe to develop models that can capture both. So this is what Steven did in his model. He first of all developed this quantitative model that can capture both reinforcement learning aspects like value reward prediction errors, but can also capture, for example, adjustments of thresholds. So this is all in one framework. And that input, so these model parameters were used as input in our joint modeling framework through an overarching group level distribution. This is the paradigm. Again, due to time and also because Steven is showing this in much more detail, I'm just going to really show you the gist here. It's very simple. People at the beginning of each trial represented a queue, and this queue was either telling them to be very fast, so you have to respond super, super quickly, or the queue told them, take your time, you have enough time, you should be accurate, emphasis on the accuracy of your response. So these are two different queues. And then in the second part of this trial, they were presented with really awkward signs. I don't think Steven invented those, but taken from other paradigms. Point is here that these stimuli in the association were connected to different reward outcomes. This basically elicited different reward schemes and also different types of prediction errors. Again, I'm just touching on it. What's important here is that we can model activation towards these cues, and we can also model activation towards these stimuli, and in particular also to the feedback phase. So we just have different onsets. And this is the model that he developed. In a nutshell, what it brings together is, as I said, model parameters from the diffusion model and also Q learning model, Q learning values. For what's D again? Stay there. D? Difference. For the difference and for the sum of the Q values. Thanks. And some error noise. Again, he's going to explain in much more detail how this is done. Most importantly, it allows us, as I said, to estimate parameters of the evidence accumulation models and also reinforcement learning. This is the model fit. For now, I'll just say the model fits the data excellently. What I'm looking at is the overlap between the model predictions, and this is the shaded areas here in all these different graphs, and the empirically collected data. What I'm looking for is that there's a tight overlap, and I think you all would agree that we see this across the board. But finally, to some results on the brain level. And here I'm showing you not whole brain images. What I'm showing you here are different nuclei in the deep brain, so based on our atlasing efforts of the deep brain. So you see just different nuclei. I'm not going into details. Basically, the basal ganglia, it's just a subset of stuff down there. And in fact, the relationship between activation in these regions. So this is all this colorful stuff here in the regions, and behavior. And what we're seeing is that not all of these regions are coding, in fact, for behavioral adjustments, but we see now a very clear, very selective involvement of a subset of basal ganglia regions coding positively with adjustments in response thresholds. So this is a general finding, first of all, of basal ganglia involvement in this type of reinforcement learning SAT experimentation. Here you see a different subset of nuclei, and what we are looking at is the thalamus. So as I told you in the beginning, right, the thalamus usually is like this block, but the thalamus is not just one block. The thalamus is super complicated. Some people think there are 10 subnuclei, some say there are 100, others say 25. So also there, they cannot agree how much it is. And I also don't know. What we're doing is we're approaching this a bit more empirically. So we basically parcellated this in as many substructures as we can clearly visualize in our data with postmortem, with in vivo and so forth. So right now we're looking at a subset of, I think, 10 substructures in the thalamus. And again, the same matrix, just focus on the rectangle. And we see that across the board, actually, the thalamus is involved nearly in all subregions coding for these response threshold adjustments. And we're still figuring out because obviously there's a huge, cross correlation between the different subparts of the thalamus. And that makes sense because they're packed so closely. So we need to find some ways to kind of normalize this or eventually correct for this. And that won't be via anatomy, given that, again, also our voxel resolution is just simply not good enough. And finally, the stratum. And again, we see here the entire stratum, we see the caudate, and we see also the putamen and a bit of the anterior part of the stratum. And again, across the board, nearly all of these substructures positively correlate with adjustments in response thresholds or behavior. And with that, I'd like to conclude, actually, given the time, that I think we're at the verge of showing more concretely that joint modeling potentially is really exciting new framework for testing brain behavior relationships. Yeah, I'm a firm believer by now, after having worked with ultra high field now for over 10 years, that this really has the potential to resolve and, in fact, overhaul many research findings and very, very prominent research findings. Also in the field of response inhibition, we have seen also different results now processing reward prediction errors. It is also a bit frightening because obviously if you find such opposing results compared to the wealth of literature, right, you really need to make sure that we're not looking at another fluke now, right, but that we're actually really improving power, improving statistical power, improving selectivity in our manipulations, so that in fact we're really be more confident that eventually this technology really resolves something novel, and maybe something contradictory, but that needs explanation. But we're working on it. And yeah, for now, this is the first study of many that are to come that we scanned, 5 or 6 different experiments that use this joint modeling framework. For now, we just show that this has high sensitivity to see selectively which regions are involved in these response setting thresholds and also reward prediction errors. And with that, I just jump through this because this is just taking too long. I definitely, of course, would like to mention the many, many people who have worked over the years on all of these aspects that I've shown you, in particular the anatomy team led by Anneke Alkemade. This is Pilou Bazin, who programmed up this automatic segmentation program and also has very, very beautiful tools to analyze fMRI data or structural data. You know this person, or we'll get to know him much better in a second. This is Nick Davidson, mathematician, and these guys all work at the intersection more on the modeling and the fMRI analysis. Of course, our many collaborators, I just listed a few here over the years, and of course also you for your attention. Thank you very much.
Okay, welcome back. And this is the second session today. We are pleased to welcome Professor Miletić. And Professor Miletić earned his PhD from the University of Amsterdam and has been an assistant professor at Leiden University for the past few years. His research focuses on understanding the structure and function of human subcortex, particularly in decision-making and learning process. He has made important contribution to our understanding of both adaptive mechanisms shape decision-making over time, combining behavioral data with neuroimaging to uncover the underlying process. In recent years, he has published a wealth of research finding in this field. Today, he will share his insight with us in a talk titled Explaining Multi-Scale Choice Dynamic. Let's welcome.
Thank you. Thank you very much for this kind introduction. And I'd also like to start by thanking the organizers and, of course, most prominently Professor Xie for inviting us here and the funding organizations that made this possible. It's been an absolute delight to be here. Beert already showed some photos of the, let's say, more cultural experiences that we've had. And I'm truly really quite grateful to be here and present my work. I'm going to just go back a little bit to this image that is from Beerte from 2010. And what Beerte has been showing mostly in her presentation is a lot of work that try to improve on the brain data side. But we want to combine it with formal modeling. And what I'm mostly going to be talking about is how we can improve things on the... You have to restart. Do I also have to restart? Oh, I'm not visible. Ah, there we go. Hello. All right. Is this better? So as I was saying, Beerte has been doing a lot of beautiful work on improving things on, let's say, the brain data side. A lot of my more recent work is on improving matters mostly on the behavioral data and on the formal modeling side of the equation. We are very much interested in decision-making, and the hallmark model, the sort of the state-of-the-art of decision-making models are these evidence accumulation models. And they've been around since, basically they started more or less at 1960, so that's really quite old literature. And they have been around for that long because they work really, really well. There's just one problem with them, a problem from the perspective of trying to understand neuroimaging data, and that is that what they do is they decompose the decision process, the decision data into the decision processes, but they don't give you a trial-by-trial estimate of these processes. You only get a block-by-block or like a condition-by-condition estimate. But for brain data, what you want is a covariate that changes on a trial-by-trial, because then you have the most powerful inference on which brain regions co-vary with these cognitive processes. This is a known problem or known issue, a known difficulty, and people have been offering different solutions for this. One is to, instead of rely on within subject reliability, on between subject reliability. So what you do is you find the cognitive, sort of, you identify for every individual what someone's cognitive process is, you quantify that, and you correlate that across individuals with brain data, like fMRI ball data. Other approaches have been to rely on statistical properties. There have been a few different models that tell you that just based on statistics, it is more likely that on a certain trial, your drift weight was higher than a different trial. That works, but the problem with all these methods is that none of these methods tell us why people train on a trial by trial. So why is it that on one trial you are maybe more cautious than another trial? Or why is it that on one trial your information processing was faster than another trial? None of these methods give you the answer to that. I've been trying to introduce a new way of answering that question, and that really starts from the very, very fundamental question of decision-making research. And that fundamental question is, why is it the case that if I give the same person the same decision problem twice, why do I get two different answers? Because these answers can differ in both, let's say, qualitatively. So one answer may be correct, another answer may be incorrect. But most prominently, they differ in response times. So sometimes people are faster and sometimes people are slower. If you would, code up a computer to do decision tasks, that would never happen. You would always get the same response at the exact same duration. And so there is something there that determines or that causes these trial-by-trial fluctuations in response times and choices. And this has been the fundamental question since 1960, basically. And the way that this has been approached is that people take their favorite decision-making task, take, for example, a random motion task. So what happens in this task is that you see this cloud of pseudo-randomly moving dots, and a few of them move coherently to the left or the right, and you need to decide what the dominant motion direction is, so left or right. Or this is an example of, let's say, a memory task where before this recognition phase, you get to see, let's say, a list of words, and during this recognition phase, you need to decide, have I seen this or not? Or a lexical decision task where you get to see a string of letters like T-R-A-Y-N, and you need to decide whether in your language that's an existing word or not. Right? So train is a word in English, trud isn't, as far as I know. And so these are pretty generic tasks. There's a wide variety of tasks that people use for decision-making research. These are just a few examples. And the dominant idea in the field is that we should analyze these recurring data, so response time data, with cognitive models. This is the most dominant one. This is the diffusion decision model, or the drift diffusion model. And, oh, thank you. What it proposes is that during a decision, you collect evidence for each choice alternative until you reach some sort of decision threshold. Right? So, for example, response A, it could be a word, and response B, or response to it is not a word. And the idea is that this is something that evolves over time. So we're talking milliseconds to seconds here. You start somewhere in between these two horizontal lines. Those are the response bounds. So you start, for example, here. And within a trial, you very quickly accumulate evidence for either it is response A or it's response B. This evidence accumulation is noisy, so that on certain trials, the noise will sort of push you to be faster to bound A. On other trials, you will also reach bound A, but you will be slower. And on other trials, again, the noise will just push you to the lower boundary, which may be, for example, incorrect. And if you do that hundreds of times, you get response time distributions and accuracy. Right? So that's the fundamental idea. You have this latent process of evidence accumulation to threshold, and if you do this often enough, you get response time distributions. This works really well. So what I've done here is I have 4 different data sets lying around, 4 different decision-making tasks. These are I think these are diffusion models that I fit to response time data. On the x-axis here is response time for these 4 different data sets. These are cumulative density functions. So these are response time distributions. You just take the cumulative, so the cumulative sum of them, except that they don't asymptote at 1, but they asymptote at the accuracy. And so if you look at the sort of where these lines end, that's a measure of the accuracy of the participants. So something like 90% in this first data set, 80% in the second, something like maybe 70% in the third, etc. And these 5 dots per line correspond to the quantile, the 10th, so this is the 10th, the 30th, the 50th, the 90th, and the 70th and 90th quantile RT. So basically this is the median response time. These lower lines are the error responses. So these are the incorrect responses. Now in all these plots, white is the data, and the green dots are the model predictions, so the model fit. And the green dots are actually quite hard to see, and that is that they overlap so tightly with the model, with the data, that it's actually kind of hard to visualize the model fit, because it just fits so extremely well. And this is sort of the standard approach to understanding response time data with evidence accumulation models. There's just one problem, And that is that if I represent the data in this way, which is the common way of representing data, it looks beautiful, right? Model fit is excellent, so we can, interpret parameters and stuff. But if I do a little trick and I transform the data, so these are the exact same data and the exact same model predictions, I see a very large misfit. So in all these plots, again, white is data, green is the model prediction. And what the trick that I applied here is just a Fourier transformation. So I treated the response times as a time series, and I tried to identify repeating patterns, repeating oscillations in these response time series. On the x-axis here is the frequency or the period of these oscillations, and on the y-axis the power, and they're both on a log scale. So what you see is that in the human data, in the white, there's this approximately 1/f power spectrum, and all these models predict a uniform power spectrum. So what does this mean? Well, it means that there are a lot of trial-by-trial contingencies. There's a lot of trial-by-trial dependencies in the response time data that I'm not fitting at all. I'm completely ignoring those. Now, this is, I think, a very interesting observation, but it's not a new observation. I certainly am not the one that discovered this. This was already shown in the late 90s. David Gilden did a lot of work on this. And this is a quote of one of his papers where he argues that the amount of variance that's associated with this temporal structure is actually a lot larger than the amount of variance that we induce in the data with our experimental manipulations. And so typically, if I have a decision-making manipulation, I'm interested in what that does to my experimental data. It does something, but it does a lot less than the natural variability that's already there just in the temporal structure that I'm ignoring. And later on, somebody pointed to me to a different paper that argues that David Gilden may have overestimated the amount of variance a little bit. He argued that it was a factor of 3 or more, but this more recent paper argues that it's approximately at the same level of experimental manipulations, right? Point is, there's a lot of variability in there that's structural, and that I think we should try to explain. So what is it? What cognitive mechanisms are there that cause this variability? So David Gilden argued that it is a signature of a complex dynamical system. So basically he said that, well, it is a nonlinear, self-organizing and multi-scale system. So it's much more of a dynamical systems approach, which may very well be true. But E.J. Wagemakers then argued that, well, that doesn't give us a formal model that we can actually work with. It's more like a signature of the type of process that's going on, rather than an actual model that, that I can work with, that I can fit to data, for example. So what E.J. Wagemakers, you've heard his name actually before in Beerte's talk, he proposed that there are a few different explanations that could cause a power spectrum that looks like the one that we observe in empirical data. One is the idea of regime switching. The idea there is that an individual participant may have multiple strategies to solve a problem, and you can alternate between them. And so in some random fashion, at some trials, you may just choose to be more accurate, and in different trials, you may choose to be more speedy, fast. And if you have different regimes that you switch between, you can get a power spectrum that looks like that. David Gilden has done some very interesting work on this in 2022. It's in PsychReview, and he modeled that with hidden Markov model. So it's definitely worthwhile if you're interested in that to have a look there. I'm going to argue for a different process, namely that, or a different cause, namely that there is a superposition of multiple short-range processes that cause this this empirical phenomenon. So that sounds kind of vague. What do I mean with that? Well, I'm going to make a very fundamental proposal, and that is that what humans do is they continuously adapt to a changing environment. Now, if you work in this field, then you may think, well, all these experiments that we do, they are static environments. They don't change. Every trial is designed to be the same. And that's absolutely true, but the argument here is that humans have evolved in a natural environment which does constantly change. And with that evolution, we may have some sort of adaptive mechanisms that still, even though the environment in a lab setting is not changing, it still may cause us to adapt to properties of the experiment that we maybe shouldn't adapt to from a rational point of view, but we do it. And that can cause trial by trial contingencies because you're constantly adapting to what you're doing and what the task is asking you to do. So that's still vague. What I'm going to propose are four different adaptation mechanisms that allow you to adapt to the task. One is that people adjust to rewards, and the other three are will come later. But let's first have a look at adjusting to rewards. This was very much inspired from the reinforcement learning literature, where people also use decision-making tasks. At least from a modeling perspective, you can argue this is just a decision-making task. You see these two abstract symbols, you need to choose between them, but based on some random property, some stochastic property, one of them is more often rewarded with something than the other. And the only way that you can figure that out is by trial and error. Right? So you start this experiment, you have no idea which of these is better in a way. You just try one, you may get rewarded, then you try another time, you may get rewarded, you try another time, and suddenly you don't get a reward. In this case, this is actually kind of fun. This is an fMRI study where they actually used juices in humans, right? So there was a very long tube where people got some sugary juice as a reward, and they needed to figure out which of the symbols were more likely to deliver juice. And in reinforcement learning, what people do is they make the assumption that people keep track of the value of each of these symbols. And so one symbol can be more valuable than the other, meaning more likely to give a reward. And these values are updated after every trial. So every time you get feedback, you update your value according to this very simple delta rule, which just says that your Q value, so the amount of worth a symbol is on the next trial, equals the current Q value plus the prediction error weighed by some free parameter. Standard reinforcement learning. And with that, you get the Q values that for both options that evolve over time. And what people do in reinforcement learning is then say, well, we take softmax, which is a nonlinear transformation of the difference in these Q values to choice probabilities. That's the standard approach there, which works in many respects, but it completely ignores response time distributions, which are, let's say, the bread and butter of evidence accumulation models. So part of the inspiration of this work was to try to combine these two sets of fundamental principles of, let's say, updating of Q values with evidence accumulation models instead of softmax. So here's how that may work. This is an example choice paradigm. You have these two abstract symbols. They're just a random font that you can pick off the internet. At the beginning of the task, you assume that people have some sort of Q value associated with each of these symbols. And now we're going to say, well, the difference in these Q values are going to drive an evidence accumulation process as formalized in, for example, the DDM. Right? So the difference in the Q value is going to drive the evidence accumulation process until you hit a threshold, for example, option A. You choose option A, you get some sort of stochastic feedback, 100 points, for example. And based on the feedback, you update your Q value that corresponds to the choice that you made. Now, the trick is that the next time that you encounter this exact same stimulus set, you have a new Q value for option A, such that which leads to a new drift rate, which leads to different behavior. Right? So it's a simple learning mechanism, but instead of softmax, we now use a drift diffusion model, which allows us to make predictions for response time distributions. I should add here that I wasn't the first one to develop this. Mats Pedersen was, I think, the original publication of the RLDDM, so that's reinforcement learning drift diffusion model. And also Lara Fontanesi has done a lot of interesting work on this. But we were also interested in this, so we, when you have the new model, you're like, well, let's just play around with it. Let's collect some data. So we did this very same simple task in some 55 participants. And what we did is we added a manipulation of choice difficulty. And difficulty in these tasks is defined by the relative probability of both choice options being rewarded. Right? If one of these options, this one, for example, has an 80% probability of being rewarded, and this one only 20%, it's pretty simple to figure that out. You don't need many trials. If this one is 60% and that one is 40%, it's quite hard. You need a lot of trials to figure out that this one is actually more rewarding than that one. Right, so that's just a difficulty manipulation. So what you then do as a modeler is you collect that data and you fit your RLDDM to it. This is some of the data that Beert also showed in the different context. It's a bit of a complex graph, but on all the x-axis here is trial number just binned into 10 bins. Right, so this is the beginning of the experiment and this is the end of the experiment. On the y-axis in the top row is choice accuracy, so the proportion of the higher rewarding choices that were given. In the middle and the bottom are response times. White is always the group average data, and the shaded areas are the 95% predictive intervals of the RLDDM, so the model fit, if you wish. I'm a Bayesian, so my error bars are always around the model and not around the data. Just that's why there's a shaded area. So the first thing to see is that people learn. The accuracy increases over trials. People pick up on which of these options are more likely to be rewarded. And the model fit area is actually quite good. Right? This is more or less on par as what softmax would predict. And so the model is able to capture the increase in accuracy over time. So that's good. But the response and distributions draw a very different picture. And just to introduce this type of plot, what I'm plotting here in these 3 lines are the 10th, the 50th, and the 90th quantile RT. Right, so this is just a histogram. If you take the quantile RTs, the 10th and the 50th and the 90th, you get an idea of the shape of the distribution. So the 50th, for example, tells you something about the central tendency, in this case, the median. The difference between the 10th and the 90th tells you something about the variance of the distribution. And if you take the difference between the 90th and the 50th and then compare that with the difference between the 50th and 90th, you get a skewness. So how asymmetrical the distribution is. So if we look at that, we see that the median is captured fairly well. And people who learn, they go faster. And that's also what the model predicts. But if you look at the model fit for the variance and the skewness, so basically the fit of the 90th and the 10th quantile, we see that the model very strongly overestimates the variance. Right? The 90th quantile is overestimated, the 10th quantile is underestimated. So that's the case also for the correct answers as well as for the incorrect answers, right? So across the board, there's an overestimation of variance and skewness. Now you can wonder, why does this matter, right? Because the median is captured well, and in most cases, we look only at median RT. Well, from a evidence accumulation model perspective, this matters a lot because the shape of the response and distribution is where a lot of information lies about what people actually do. And if you're not capturing the shape of the response and distribution in evidence accumulation modeling world, you're not doing a very good job because that's more or less what these models are designed for. So at this point, we were disappointed because, because of this misfit. And what we set to do is try to improve this by by basically going back to the drawing board. Because what we've done so far was just to take the very simple delta rule, let's say the most standard version of reinforcement learning that exists, with one of the most standard versions of evidence accumulation models that exists. And by doing that, we ignored the wide variety of models that have been developed over the last 60 years in both fields. Right? And this wide variety of different, let's say, modeling options gives you very large combinatorial explosions of potential RL-EAMs, right? Because you can combine every one of these reinforcement learning models with every one of these evidence accumulation models, which gives this, exciting new field, but at the same time also quite difficult new field, because where do you start? What do you do? So what we did was basically keeping the reinforcement learning part the same and building more on the evidence accumulation modeling side. And the reason was that the evidence accumulation models are the ones that account for the response and distributions, and that's where the misfit was. So this is just a depiction of the RLDDM again, where you have a two-sided evidence accumulation process with two bounds. But you can generalize this to a race model, where you say that, well, for every choice option, there's one accumulator. And they accumulate the evidence for their choice that they represent, for that symbol. And whichever one reaches its bound first, that's the winner. It's a race. And in this very simple setup, what we did is we said, well, the drift rate for every evidence accumulator is simply a weight version of the Q value plus a shared intercept. Right? So one racer, one Q value, and then it's a race. We also took a more complex version of the racing diffusion model, the advantage race, which still has just 2 evidence accumulators, but we made a little trick where we said that instead of every accumulator just accumulating evidence for one choice option, it's going to accumulate the advantage of one choice option over the other. And so instead of absolute values, what we care about is how much better one value is compared to the other one. And that seems like a small change, but it has a very large impact. Right, so we fit all these models to this same experiment. On the left is the RLDDM. This is what you've already seen before with the misfit on the response time side. And in the middle is the RLRD. So that's, let's say, the simple version of the racing diffusion model. What you see here is that the response time distributions of the correct and the incorrect responses are already captured a lot better. As in, the variance is no longer overestimated. Skewness seems to be fine. It's just a little bit of an overestimation of response times at the beginning of the experiment. Same goes for the error RTs, but this comes at the cost of accuracy, right? So the accuracy curve here is strongly underestimated. So what this model then tells us this, well, it looks like if people would do this, you would indeed expect the same response times and same response time distributions, but you would never get to the accuracy that you observe empirically. And the RLARD combines more or less the best of both worlds. You see that the fit of the accurate responses and the error responses are almost perfect. Also, this overestimation of the first trial bin is solved. Plus, it fits the accuracy curve about as well as the RLDDM. It's slightly worse, but it's at least all within 95% credible interval. In the interest of time, I'm going to skip a few things. I just want to give you a little bit of a flavor of where I'm going to bring this next. Because this proposal so far that humans continuously adapt to the changing environment, you can argue that that's the case in when there's rewards. But in many of these tasks that I showed before, there are no rewards. So how could that then work in this type of task? Well, there may not be rewards, but it could still be the case that people adapt to certain properties of the experiment. So one is, for example, that they adjust to the perceived probability of events. So again, statistics, but not the reward statistics, but more the statistics of what actually happens during the task. So this is just an example. So suppose that on trial 1, the stimulus tells you that the left answer should be the correct answer, right? So your random motion task tells you it should be left. Then this is a little sort of an animation of the race between the 2 accumulators, and the left accumulator wins, so you respond left. Now we're going to assume again that there's a Q value, and this Q value now stands for the relative probability of left-right. And it's bounded between -1 and 1. It's not a probability, but it's more like sort of this general thing that co-varies with left and right. Now, because the first stimulus was left, we update the Q value for the second trial, and we make the Q value a little bit more left. So in this case, 1. And that Q value is going to influence the relative height of these response bounds, such that we have a bias towards saying left compared to saying right. And again, we have this evidence accumulation process. On the second trial, the stimulus was left again. The left racer wins. You update your Q value again with left. Your third Q value is going to be higher, which means that the difference between these response bounds is going to be even higher in the next trial. And perhaps due to this bias, on the third trial here, a mistake is made because the stimulus tells you it should be right, but this left accumulator actually wins, right? So the left response is the response we observe. And again, you update your Q value, but now the reward isn't 1, but it's a -1. So that's your Q value now is going to decrease and your bias is going to be moved towards the other side. Right, so why bother? What's the point of all this? Well, what this allows you to do is explain certain sequential effects that are present in the data. Specifically, what we call stimulus history effects. Again, a bit of a complex graph, but here on the left, we see, or here on the x-axis, we see the stimulus of the stimuli presented on the previous 3 trials. So, so on the far left here, the previous 3 stimuli were left. Here, the previous 3 stimuli were right. And here on the y-axis is the probability of choosing left after these 3 previous stimuli, for example. So in case the previous 3 stimuli were left, then the probability of choosing left of people is about 57%. And it's quite far over the 50%, which it actually should be, because these are completely randomly drawn. If the previous 3 stimuli were right, then the probability of somebody choosing left is some 42% or something like that. So what you see is that participant behavior can be predicted pretty well based on just the previous stimuli that were shown. The crosses here are data and the error bars are model fit again. So this model, which incorporates this adaptation mechanism, allows the capturing of these sequential effects that are pretty strongly present. In the top row, we see sort of the same idea, just now in a response time domain, where the colors here correspond to the 2 different choices. So white here is the left choice. And what this says is that if the previous stimuli were left and you choose left, then that was a pretty fast choice. If the previous 3 stimuli were left and you choose right, that probably took you more time. You see this sort of crossover pattern. And that's not just the case in this third data set, but in all these 4 data sets that I introduced in the beginning. Right, so this is just intended to give you a flavor. Other things that people can adapt to is the perceived accuracy of their own behavior. Right? So people can keep track of how well they're doing and then adjust their parameters accordingly. Right? If you're making a mistake, you should change something. And a final thing that people can adapt to is how difficult the task is. Right? If they perceive it to be very difficult, perhaps you should be more cautious and slow down a little bit. But if it becomes harder, it becomes easier, you can speed up, right? It's easy, so you can just be faster, it will be fine. But in the interest of time, I'm going to skip all this beautiful work. Just very briefly, what I wanted to show is this is what I started out with, right? These Fourier spectra showing the misfit between what the model predicts and what people do. What I wanted to end with is what these models then give us in terms of explanations for this power spectra. And it's depicted here. And what it says is, so first of all, it's definitely not perfect. I'm not going to claim that I've explained the entire power spectra, but I think we are actually coming quite a long way, and mostly in, let's say, the faster fluctuations. So the fluctuations of up to, let's say, 5 minutes of time, which is like a block level of experimental trials. In a slower domain, so 5 minutes and slower, up to say 30 minutes, there is a lot of variance left. Unaccounted for, which may have to do with processes like mind wandering. People maybe just stop caring at some point. They get distracted. Something like that may happen, but that's all sort of speculation at this point. And with that, I'd like to summarize this. What I hope to do here is sort of, instead of focusing on these specific empirical phenomena, really introduce this as a general modeling framework, how we can incorporate these learning mechanisms, these adaptive mechanisms in these evidence accumulation models, and then try to understand these temporal dependencies that are clearly and very strongly present in empirical data. And at the same time, acknowledge that there's definitely a lot of variance still left to explain, which basically means that I don't know what it is, but it's there and it's strong. So it's worthwhile looking into that. Then the most important slides, the thank you notice, definitely, of course, to Beert, who has supervised me in the past. We're colleagues now, but we still work very closely together. And a lot of other people who have worked on this lab. Andrew is also closely involved with a lot of this work. And you for your attention. Thank you.
We still have some time. And is there any question?
Still a very interesting talk. So I just want to learn more about the race diffusion model. So could you explain a bit more about this model? And yeah, so my question would be, do you have any assumption about the independency between the two parallel processes? And also curious about when you combining the reinforcement learning model and the choice decision-making models, how do you expect these inter-trial effects would affect on the criterion shift, or just only focus on the drift diffusion drift rates?
Thank you. These are, sorry? Yeah, no, excellent questions. Thank you so much. So as with respect to your second question, unfortunately, I don't think I really have time to explain that, but those are really ingrained in these latter two mechanisms, which allow for adjusting response bounds, so response caution as well as urgency. In the interest of time, it is fully and obviously greatly explained in this preprint. No, we recently published this online, so there is a lot of more information in there if you're interested in that. It's the last one. It's on OSF and PsycArXiv. But as for your first question, the independence between accumulators, that is a very long-standing question indeed. To what extent accumulators or racers are truly independent? There are model variants that have some sort of latent inhibition in there. The leaky competing accumulating model is the LCA is the most famous one. You could argue that the DDM is a model that incorporates what we call feed-forward inhibition. So it's let's say the input here is the difference between Q values. So you could say, well, you know, what we're looking at is not inhibition at the accumulator level, but it's already inhibition at the Q value level. They inhibit each other, and the only thing that you integrate is their difference, so the result of that. There's also other variants with feed-forward inhibition. The advantage race also incorporates that idea of feed-forward inhibition by also integrating the difference in Q values. But that said, there is also models indeed that do latent inhibition. So these two accumulators really basically put each other down. The reason I haven't been doing that is that it's more a technical reason, and that is that these models have no known analytical likelihood function, which means that if I want to fit these models to empirical data, for every trial, I will need to fit tens of thousands of trials to approximate the likelihood, which makes it practically almost impossible just computationally to fit it to data. So it's partly sort of a pragmatic choice here that I just cannot really estimate it. It's impossible. But it's a good question because it's quite likely that there is some sort of processing there going on, some sort of competition.
Thank you very much. And my question is from Professor Forstmann. Okay, so as you mentioned about like the black hole, the subcortical region. So I'm just curious about like how it correlates to our current understanding of like from the developmental perspective, like the diencephalon, and then how it turns into all those structures. And the second question is the number 455. Like, is it like fixed, or it would depend on like age or something? Like maybe it's absent in like children, so it can be like something like a vestigial, something like that.
Yeah,
Thank
great.
you.
Thank you very much for this fantastic question, which I think I'm really not able to answer fully because I haven't looked into the developmental trajectory really. My hunch is that at birth, all of these structures are developed, given that they are developing in a very early stage, given that also we see these systems, you know, in much kind of simpler organisms, such as, for example, rodents, certainly fewer, and I can't put my finger on a certain number, right? I don't know exactly, because the homologies are also not entirely clear, right? It's not as if there's a red nucleus in, you know, every mice species. But one thing I can say is that they develop very early on during gestation, much earlier than, in fact, actually the whole formation of cortex, and that these are systems that are fundamental also to for very basic functions of the human body, meaning regulating homeostasis, regulating temperature, hunger, sleep-wake cycles. So these systems need to be already in place in order, you know, to make you live, if you want. Whether they change just in its appearance, let's say more at the end of age, right, or over the adult lifespan, we haven't seen that. We have done one study, in fact, also Steven has done that, where he, in fact, looked into the changes of myelination and iron concentrations, for example, in this database that I presented. So from 18 years onwards to 80 years. And we see changes, but we do not see changes that certain structures all of a sudden disappear or that they join, or, you know, like the entire morphometry, in fact, alters. Obviously, there are changes that are coming with aging. For example, enlargement of ventricles. We also see enlargement of the spaces between gyri, but still not to an extent that really structures completely, you know, disappear or go down. That may very well be different in diseases such as neurodegenerative diseases such as Alzheimer's. We're currently starting to collect data of these patients. So to be continued, really, to be looked into. But I would think that you know, the set of 155, and this is arguable, right? We counted based on a book that is devised by neuroanatomists in the world. So they came together and they wrote this book, and they basically created this nomenclature of all these structures. It's not a number that I just kind of, you know, vaguely thought of, but basically just counted in this book how many are there. And this will include, or this does include also, in fact, layer specific information. So whether you call this a nucleus or not is also open for discussion. But I think one thing can be said for sure is that eventually you see many more structures or different, you know, structural entities deep in the brain compared to cortex. And again, these will be developed very early on in also in childhood. Yeah.
Okay, it's about time, so we'll close the seminar today. And let's thanks again for, okay.